Good morning, students. In previous lecture, we discussed about the complete mechanism of muscle contraction, the sliding theory of muscle contraction we have discussed. In that sliding theory, we had discussed that energy is provided by ATP, and ATPase enzyme breaks the ATP into ATP and phosphate. Today, we are discussing here about Cori cycle. The energy requirement in the intense activity is fulfilled by Cori cycle. Myself, Dr. Ashok Kumar Siraya, Associate Professor, Department of Zoology, SPC Government College, Ajmer. As we discussed that during relaxed period, energy is used from oxidative phosphorylation. But during intense activity, this energy don't fulfill the requirement of muscle contraction. For this extra requirement, ATP is formed by anaerobic glycolysis. Oxidative phosphorylation creates more energy than anaerobic glycolysis. In this process of anaerobic glycolysis, lactic acid is formed as byproduct. This production of lactic acid starts to accumulate in muscles. Lactic acid from glycolytic system will diffuse into bloodstream, then transported to the liver. The lactic acid reserve or accumulate in a muscle, and this lactic acid by the mean of glycolytic system diffused in bloodstream and ultimately transported to liver. In the liver, it is converted back to glucose from lactic acid or lactate, which is then available to muscle for energy. Means during intense activity, glucose converts in lactic acid transported to the liver. In liver, it converts back in glucose and again available to muscle for energy. This conversion of non-glucogenic materials in glucose is known as gluconeogenesis. Means there is a process in the liver named gluconeogenesis where non-glucogenic materials like lactic acid converts in glucose. The conversion of glycogen in glucose is known as glycogenolysis. If we break down the glycogen into glucose, it is known as glycogenolysis. And if glucose is formed or constructed by non-glucogenic material, it is known as gluconeogenesis. And vice versa, if glucose converts into glycogen, then this process is called glycogenesis. Glycogenesis means glycogen formation. And the conversion of glucose in glycogen is known as glycogenesis. This cycle, conversion of glucose, lactic acid, and glycogen occurs during intense activity of muscle, is first described by scientist Corey and Corey. For this cycle is known as Corey cycle, or because of the lactic acid present in this cycle, it is known as lactic acid cycle. Means we can say it Corey cycle or lactic acid cycle. If we see the line diagram of Corey cycle in muscle, there is glucose molecule which convert in 2-pyruvate. If oxygen present, it undergoes in oxidative phosphorylation, and if the oxygen is not present or less oxygen is there, then it converts in lactate. Means one glucose molecule converts in 2 lactate molecule or lactic acid molecule. Then it transfer via blood to the liver. In the liver, 2 lactic acid converts into pyruvate. It is a reversible reaction. Lactate to pyruvate and pyruvate to lactate. This process, this is anaerobic glycolysis where glucose converts into lactate, and this process, when lactate converts in glucose, it is gluconeogenesis. Then lactate converts into pyruvate, 2-pyruvate make glucose. Thus, the entire cycle, glucose to lactate, lactate to glucose, runs during the intense activity of the muscle contraction. In the liver, if the glucose is in a less quantity, the glycogen supply the glucose by the process of glycogenolysis, means glycogen breakdown and formation of glucose. And the lactate, which are in extra quantity, converts in glycogen by converting it glucose, and then glucose vice versa convert in glycogen. That is glycogenesis. Thus, the Cori cycle provide the energy during intense muscle contraction. Thank you. In next lecture, we will discuss about energy source in muscle contraction and how the radially energy provided by the ATP and uh, how ATP is created from creatine phosphate. We will discuss in detail in next lecture. Thank you.